Hello to our friends joining us via recording. We are starting our discussion of the last lecture lesson for this semester, and this is the lecture lesson about the special senses. So to get us ready for this lesson, we split into groups and we tried to remind ourselves of what the various special senses are and what the general senses are. We talked a little bit about special senses in lab, both in the lab packet, as well as when we were talking about cranial nerves. So as we go through and identify special senses, we're also going to remind ourselves of the cranial nerves that we use to collect that kind of sensory information. So we're going to have a little bit of a free-for-all here for my friends who are here in class today. Up above my finger, there is a pencil. Go ahead and click that pencil. I'm going to give us here about a minute. Let's go through and circle for each of these types of sensations that you see here listed. Let's circle whether they are a special sense or a general sense. So go ahead and grab that pencil and let's see what we came up with when we were in our groups. For a while there, my friend with pink was just going to town. Excellent. Looks like we agreed with each other, so that's a good thing. And we're happy about it, too. Look at us. It's the Red Bull and the coffee this morning, right? For my friends who came in late, I found a little meme online that said that I put Red Bull in my coffee instead of water, and now I can see noises. So we're feeling good. Okay, so let's talk about this. Like I said, we generally agree. Look at our fun little person here. So let's start with balance. We agree that balance is an example of a special sense. Now, in anatomy, our special anatomy word for balance is equilibrium. When we talked about it in lab, we used that word equilibrium for it. There was a cranial nerve that helped us out with equilibrium, or it helped us out with balance. What was the number and the name of the cranial nerve that helps us with equilibrium? Yeah, so it's cranial nerve number 8. That's its number. And its name is the big long one, vestibulocochlear. You all spelled it right. Let's see if I can spell it right. Vestibulocochlear. Cranial nerve number 8. The vestibulo part of its name is the part of the cranial nerve that helps us with equilibrium. Hey, the second part of its name, the cochlear part of its name, actually helps us with our next special sense on the list here, and that's the sense of hearing. So cranial nerve number 8, the vestibulocochlear nerve, also helps us out with the process of hearing. So 2 special senses done by the vestibulocochlear nerve, cranial nerve number 8. We agree that the sensation of pain is an example of a general sense. I've said this a few times in office hours, but does anyone happen to remember when you detect pain, what are you actually detecting? Does anyone remember what I usually say? What pain actually is? Okay, yeah, so it can be caused by pressure. It could be caused by temperature too. When we talk about pain, it's actually detecting all of these various general senses that we talked about. So things like stretching, things like temperature, texture, pressure, any of your special senses, if you detect too much of it, that actually registers as pain. So if something is too hot or too cold, your body registers that as pain. If you get too stretched out, your body registers that as pain. If you're holding something or putting your body weight, say, on a Lego or a nail, when we have too much pressure, that's detected as pain. So when we get a lot of a type of general sensory information, too much where we kind of overwhelm that general sense, it starts to send a new message to the brain. Not only is it telling the brain, I detect that there's a temperature thing going on here, we're sending the message there's too much of the temperature thing. Or we're sending the message that we're too far stretched out, bend that leg back. Pain is when I detect too much of one of my other sensory informations. Yeah, so like Christina said, too much sensory information, too much stretching, too much temperature, too much pressure, too much of anything, that's what your body calls pain. Someone said beauty. We detect beauty as pain. To be beautiful, especially these days, right? That's pretty painful. It takes a lot. So I don't disagree. Okay, so we mentioned these ones since we're talking about pain. We mentioned that stretching is general sensory information. We mentioned that the temperature of things is general sensory information. Texture is general sensory information. That means our other things on the list here, like smell and taste and vision, those are all special senses. When we talk about the sense of smell, which cranial nerve helps us with the sense of smell? Yeah, cranial nerve number 1 is called the olfactory nerve. My special anatomy word for the process of smell is called olfaction. Olfaction is the process of smell. So the olfactory nerve, cranial nerve number 1, helps us to do smell. When we talk about the process of taste, the technical anatomy word for taste is called gustation. Gustation. When we talk about taste, there were a couple of cranial nerves that we mentioned that help us out with taste. Did we remember what those cranial nerves were that helped us with taste? I think we even briefly talked about it in our lecture sometime this week. Yeah, so one of them is the facial nerve. Cranial nerve number 7 is the facial nerve. That's one of them that helps me out with the process of taste. Yeah, and the other one is cranial nerve number 9. Cranial nerve number 9 is called the glossopharyngeal nerve. Glossopharyngeal. Glosso means tongue. Pharyngeal means pharynx or throat. So some of the biggest places we taste in our body is with our tongue, and in the very first part of our throat, we actually have some taste buds up there. So cranial nerves number 7 and number 9, the facial nerve and the glossopharyngeal nerve, these two help us with taste. Yeah, so Nicole asked a good question for us. She asked about, let me put it here on my slide for my friends on the recording, cranial nerve number 12. What was the name of cranial nerve number 12? Yeah, cranial nerve number 12 is called the hypoglossal nerve. Hypoglossal. It still has that tongue part in it, that glossal part, but the hypoglossal nerve is what we call a motor nerve. It only sends motor information. It doesn't collect any sensory information. So the hypoglossal nerve is going to help us out with tongue movement. Tongue movement. So it's going to help you, for example, when you have food in your mouth and you're moving it around in your mouth, it would help you with that, but it does not help you with tasting. So even though we've got the tongue thing in here, it's not going to help you out with tasting. That's just going to move the tongue. The one that actually helps us to taste, glossopharyngeal. Glossopharyngeal. All right, last special sense, the one we're going to spend our time on today, is the special sense of vision. And which cranial nerve helps me with the special sense of vision? Who helps me to see? Yeah, cranial nerve number 2, the optic nerve. Hey, I ask you this question all the time, so surely we can rattle these off. There are 3 cranial nerves that help you to move your eyeball. What are the 3 cranial nerves that help us with eyeball movement? Yeah, so number 3, number 4, number 6, they all help me to move. So number 3 is what we call the oculomotor nerve, eyeball movement. Number 4 is that trochlear nerve, also helping me with movement. And number 6 was that one called abducens. So all of these are eyeball movers. They don't help me to actually see. They move the eyeball, but they don't help me see. The only one that helps me see is cranial nerve number 2. Here's a wink, wink, nudge, nudge, or not even that subtle as a wink, wink, nudge, nudge. We just need to make sure that we know which cranial nerves are involved in each of these special senses. We covered cranial nerves in lab, and they're coming back to haunt us again in lecture. If we see them in more than one place, you will see them on the exam. So make sure we know cranial nerve number 8 helps me with balance and hearing, equilibrium and hearing. Make sure that we know that number 1 helps me with smell. Make sure that we know that 7 and 9 help us with taste, and number 2 helps us with vision. So there's our cranial nerves doing special senses, and we figured out what those general senses were. Then we switch back to the brain. When we talk about the brain, remember that special sensory information all gets to go to its own special place in the brain. We process it in particular areas of the brain. So let me do a quick reminder for us here. Our things that were special sensory information, I'm going to underline them in blue, my special senses in blue. Everything else is general sensory information. When I talk about general sensory information, all of that goes to the same place. I outline my red stuff here. When we talk about general sensory information, where is all of that sent in the body? Did we figure that out? Yeah, so it's all going to the parietal lobe. In particular, there is a map somewhere in the parietal lobe. Where's that map where we like to send the information? Yeah, so we've got a map called the sensory homunculus. The sensory homunculus. It's inside the parietal lobe. In particular, it's in this bump right here. This bump right here called the post-central gyrus, the bump that divides up the frontal lobe from the parietal lobe. That's where I have that sensory map. And that sensory map is where I send this general sensory information. So if we're looking at a lobe, if we're trying to figure out which lobe I send the information to, big picture, I send it to the parietal lobe. That's where I send my general sensory information. More specifically though, it's that thing called the post-central gyrus. The post-central gyrus, where I have the sensory homunculus. So pain information, stretching information, temperature and texture, all of that kind of stuff is going to that map that we have along the post-central gyrus. When we talk about our other special senses, they go to various lobes in the brain. So let me flip our question on its head a little bit. I'm going to point to a place in the brain. You tell me what information goes there. When I'm back here in the occipital lobe, what kind of information do I send to the occipital lobe? Yeah, that's my vision information. Vision in the occipital lobe. When I'm here in the temporal lobe, technically there are two kinds of information that go to the temporal lobe. Yeah, so the first one is the one that makes sense, right? Because we're right by the ear. That's where hearing information goes. The second one is a little bit weird. The second one is smell. Olfactory information is also processed in the temporal lobe as well. When I talk about gustatory information, taste information, where does that information go? Yeah, so we're mentioning the name of where it goes. Taste information goes to something called the insula. Where is the insula? Where do I find this? Yeah, so the insula is a deep lobe. It's a lobe of the brain that's actually kind of underneath the frontal lobe and the parietal lobe and the temporal lobe. If we dove kind of right in here, if we moved everybody else aside, deep down underneath here, we would have a lobe called the insula. The insula. So the insula, which is deep, that lobe of the brain is where I send taste information. Taste information. Nicole asked if we labeled it invisible body. I don't know if we did or not, actually. It's a little bit challenging to isolate it by itself. Just because it is deep underneath. I do have one lab model that shows it, but obviously we're not in the lab for me to show that to you. So I'm thinking you probably didn't actually label it. I think that was one we just wanted you to know what it does. And the big thing that it does for the sake of our class is when we're talking about taste, the process of tasting. So the one other special sense that we haven't labeled from our list is the special sense of balance. And this one is a little bit funky. This one is a little less centralized than some of my other special senses. Help me out in the chat. What were some of the places that you said balance information went? Yeah, so it looks like most of my group said what I would probably say is the correct answer as well. The cerebellum. Remember when we were talking about I'm labeling it the cerebellum. That's not helpful. When we were talking about the parts of the brain, we said that your cerebellum was really important for equilibrium and balancing. So this is the part of your brain that checks out what's going on with your movements and make sure you're doing what you want to do. It's making sure you're not falling over. Those are the kinds of things that we do with the cerebellum. So yeah, I would say the best answer for where that balance information goes is in the cerebellum itself. Not technically a lobe of the cerebrum, which is why, full disclosure, I probably won't ask you about that one on the exam. I'm much more likely to ask you where I do vision, where I do hearing and smell, where I do taste. So make sure we know those. Doesn't hurt to know that the cerebellum helps us out with balance. I'm less likely to ask you that though. Before we move into the process of vision, any questions that we have right now? Or shoot me an emoji. What are we thinking? What are we feeling? Here's my daily penguin. Y'all got to remind me at the end. I need to draw you a penguin again. It's been a little while. I like it. I am never going to see Mushu the same again, Hannah. I'm so happy that you found him. I'm liking the owl. All right, well, let's go to, let me pull up my picture here. Let's go here. Yeah, Mushu must never be forgotten. I haven't actually watched the new Mulan, but I'm like really sad that Mushu's not there. Has anyone actually watched it? I know you could stream it on Disney Plus. But let's be real, I've got 2 little kids at home, so it's not worth $30 to watch it. Yeah, Nicole's right. Yeah, Hannah's boycotting it because there's no Mushu. Yeah, maybe someday I'll watch it, but man, it was like iconic in my childhood. Fun fact, bunny trail tangent here. I have 3 younger brothers, and we literally used to argue over who got to be Mulan when we played Mulan in the backyard. Like, that's how iconic it was in my childhood. Granted, I was the only girl, but we argued over who got to be Mulan. So that's fun times. Okay, well, now that I've rambled too much, right? Let's talk about the process of vision. When we talk about vision, or your ability to see things, what you're actually perceiving and seeing are light waves. So I promise I'm not going to ask you about this on the exam, but I wanted to show you to help you understand when we talk about seeing things in color or seeing things in general. What you're seeing is the light that bounces off of something. So light travels at different rates. Some light travels really slowly, like I see here with red light. Some light travels more quickly, like we see with blue and purple. When you perceive different colors, it's because you detect how quickly the light is moving. This is why, by the way, people rail against blue light or purple light coming from devices. They're not wrong about that messing with your circadian rhythms, because way back in the day, you know, pre-technology, when humans were living in Little House on the Prairie, the only time they saw blue light was when the sun was out. So our body clocks are trained to, whenever we perceive light that travels this quickly in blue light or purple light, it's perceived to tell us that the sun's out, to wake us up. When we talk about things like red and orange light or yellow light, this is kind of the lower energy light. That kind of light, we see that in a candle. We see that in a fire. That kind of light helps us to kind of slow our body down. But when people talk about the dangers of blue light, they're not wrong. Circadian rhythms can really get thrown off when we're seeing this high energy light. So when you're perceiving color, the perception of color has to do with how quickly those light waves are moving. And again, what color you see comes from which light waves bounce off. When we see something that's white, it means that all of the light waves are coming back toward us. We see all of them. When we see something that's black, it means that none of the light waves are bouncing back. So this is why when you're outside in the Texas summer, you don't want to wear black, because you're going to absorb all of the light waves from the sunlight in your clothes. So what we're seeing is the light waves that bounce back. Your eyes detect how quickly or slowly they're going. When we talk about your eyes, and the good news is we've done this in lab already, so we know about these eye parts here. When you talk about your eyes, there are 2 kinds of structures in the eyes. Some of those structures are called visual structures. Visual structures are the ones that are directly involved in vision. They actually interact with the light waves. The rest of them are called accessory structures. Accessory structures. And these are ones that really their job in life is to keep the eyeball healthy, to keep the eyeball happy. When we talk about visual structures, kind of the best way to figure out what the visual structures are is to imagine that you are a light wave traveling through space, traveling through an eye. What would you bump into, or what would affect your ability to be perceived? So let's pretend that we're a light wave here. We're coming in. The first structure that we hit on the eyeball is the clear outer part called the cornea. The cornea, the clear outer part of your eye, is actually probably the most important part for altering the direction that your light waves are traveling. So The first way we perceive light from all over the place in the environment, the cornea helps to focus it on our opening in the eye called the pupil. So the pupil is just an open space in the middle of your colored iris in the eye. Depending on how much light is coming from the environment, the pupil can get bigger or smaller. The amount of open space gets bigger or smaller. When the light waves pass through the pupil, they bump into the structure called the lens. And the lens is the second structure in your eye that can also bend light. The job of the lens is to make sure that we bend our light waves to focus them all the way back here on this special spot in the eye. And in lab, we called it the Macula Ludia. The Macula Ludia. Does anyone happen to remember why we cared about the Macula Ludia or what was going on with that? I know we've all done the lab mind dump, right? Yeah, Nicole is right. The Macula Ludia, this is the place in the back of your eye where you can see most clearly, the Macula Ludia. So it's the place where your lens is trying to focus light onto. We can only see light when it's actually focused on the retina of the eyeball. You've got a lot of light that comes in that never makes it to any part of the retina. If we don't get focused on the retina, we can't see it. The place in the retina where you can see the clearest is a place called the Macula Ludia. And this is the place in the eye where we have the most of a type of cell called cones. Cones. We're going to talk about how your retina has two types of cells to perceive light. It has rods and it has cones. The Macula Ludia has a whole bunch of cones, tons and tons of cones. So these are cells that see really clearly. They see really sharp. We put a whole bunch of them here in the Macula Ludia. We can see really clearly here. So to answer your question, Christina, the retina is the name of all of the nervous tissue that can perceive light. It's made of rods and cones. The Macula Ludia is the place in the retina where we have the most cones, or it's the place where we see the clearest. What helps us to focus the light on the retina and on the Macula Ludia in general is the lens. So the lens will focus the light. Most of it we try to focus right here on the Macula Ludia. If we end up anywhere else on the retina too, we can perceive it. We just can't see it quite as clearly. So here's where you see clearest. But if you think about, there's that term peripheral vision, the stuff that you kind of see out of the corner of your eye, that's all this retina stuff up here. So a peripheral vision is all the rest of the retina, and the Macula Ludia is what you see straight on when you're staring at something, what you're focusing on. Remember from lab that we talked about how there's two kinds of fluid in the eyeball. These fluids are called humors. So the vitreous humor is here in the back of the eye, behind the pupil, behind the lens. And then we have the aqueous humor here in the front. The fun fact about the aqueous humor is that over the course of our class time together, you will completely replace all of your aqueous humor. That's how quickly we make it. We don't really change out the vitreous humor. And for any of my friends who have what are called floaters, where there's little gray spots in your vision that kind of float around, this is where those floaters are found, inside the vitreous humor. I don't know if I've got anyone else in class today that has those, but man, I hate my floaters. It's little debris, or a lot of times it's proteins that are in this vitreous humor here. When it clumps up, it gets between those light waves and your retina and basically makes a little shadow in your vision. So those are things, if you're interested in it, they're called vitreal floaters. Vitreal floaters. Nicole says she grew out of hers. That's awesome. I want to grow out of mine. Yeah, Christina asked if you can get them corrected. I literally actually did a Google search recently about it because I'm spending so much time on the computer and I see them a lot with the computer. They don't typically deal with them because it's risky. The main way I was reading about that you can deal with vitreal floaters is they can actually remove the vitreal fluid and put in new vitreal fluid to get rid of the floaters that are inside of it. But if you think about what's going on, part of the job of the vitreous humor back here is to keep your eye at the right shape and pressure. So it's really risky to some extent to remove all of that fluid and then put new fluid back in there. So it's not something that they do very often. There has to be like substantial problems for them to do that. Yeah, well, Christina says she'll just wait to grow up then. Yeah, we all will. Yes, let's hope that we grow out of our vitreal floaters. Yeah. Okay, so let's talk about how vision works. Right now, you probably are kind of going back and forth between focusing on two different things. You're probably focusing on your computer screen that's maybe a little bit far away, and then you're probably looking up close at your notes or at something you're writing on, or based on the conversation we were having before we turned on the recording today, we might have our phones out, a couple of us, right? Put those phones away. We're looking at stuff up close. When you go from looking at your phone up close to looking at your screen far away, the way that we are able to focus on both of those things clearly is because we're changing the shape of the lens. So when we're perceiving something that's up close, like this letter R up close here, notice that our light waves come into the eyeball. They come in kind of at a wide angle because we're so close to what we're looking at. Remember that the cornea starts to bend that light a little bit and puts it onto the lens. Now, the angle that these light waves are coming into the lens actually has to compress down. So see my little arrows here? My lens is bulging, or it's compressing down, making the lens fatter, or as I like to say, we're doing the little short and stout teapot right here. So we squeeze that lens down short and stout. That focuses those light waves on the macula ludia, on the retina itself. If we're not looking at something up close, though, that the angle is coming in so hard and so fast, what the lens will actually do is it will stretch out. So we're flattening it more long and lean, if you will, if we're looking at something that's far away. When the light comes from far away, by stretching this lens out, we still end up focusing it on the macula ludia in the back of the eye. But even though the light waves came in at a different angle, I changed them to end up in the same place. The big idea with distance vision and close vision is we have to change the shape of the lens to be able to do that. To make your lens short and stout, to squeeze it down together, we use what are called the ciliary muscles, or in lab, I believe we call them the ciliary body. So when we squeeze those muscles, we essentially kind of push down or pull together those muscles to make this lens really wide. When the muscles release, this lens kind of relaxes. So when they're not contracting down, the lens gets bigger. When they are contracting, it makes the lens a little bit wider, or makes it do the short and stout thing. But here's something to keep in mind with all kinds of vision, because we're going to talk about problems with vision now. Here's underline, highlight, star idea, and I'll type it for us. For you to be able to see something, the light waves, bear with me, the light waves must focus on the retina. For you to be able to see something, the light waves must focus on the retina. Regardless of whether those light waves came from really far away or whether those light waves came from really close, we've got to get them to the retina. If they don't make it to the retina, we can't see them. So if we somehow focus our light waves here in the middle of the vitreous humor, hey, I promise this is not a trick question. If I focus my light waves in the vitreous humor, are there rods and cones? Are there vision cells in the vitreous humor? What do we think? Yeah, the vitreous humor is soup. It's the stuff we studied in lab. It's liquid. Because it's liquid, there's no rods and cones in it. If I focus my light right here, there's no rods and cones to see it. I can't see light that gets focused right here. If I focus my light back here, behind the eyeball, again, there's no rods and cones back here to see it. So I've got to get my light focused on the retina. That's why your lens changes shape. That's why the ciliary muscles or the ciliary bodies either will relax when we're seeing something that's far away, or they'll contract when we're seeing something up close, because that will allow them to bend the light waves in a way so that you would get them focused on the retina. So keep in mind as we start talking about issues with the process of vision, it all comes down to this. I've got to get my light focused on the retina. If I mess up where my retina is, if I mess up how I focus my light, doing any of those things is going to make it so that I can't see clearly. Let's look at our first vision disorder. Actually, our first two vision disorders. Question first, light gets focused on the retina. No, so Christina, light gets focused on the retina by the lens. The lens focuses the light onto the retina. The part of the retina that the lens wants to focus the light on is the macula ludia. So the goal of the lens is to focus light onto the macula ludia. That's one part of the retina. It's the part of the retina where you can see clearest. So the entire inside of the eye is lined with a layer of nervous tissue cells called the retina. The one place in the retina where you can see the clearest is the macula ludia. So it's a specific location on the retina where you see the most clearly. Yeah, so it's the clearest spot. All of the retina we can see on, but we can see best at the macula ludia. Okay, so, oh, no worries, Christina. That's a good clarification for us. We're using not English words, right? So it's always good for us to figure out what the not English words mean. Let's talk about the two most common eye disorders. The first eye disorder is called myopia, or it's a myopic eye. Before I give you the easy person word for it, does anyone here in class today know that they have myopia? I very much have myopia. Yeah, Christina knows she's got myopia. Anybody else? Nicole too. Yep, so here, let me give you the normal person word or the easy person word for it, and we'll see if any else of us realize we have it too. Myopia is also known as nearsightedness. Nearsightedness. Anyone else got myopia now that we call it nearsightedness? So nearsightedness means that we can see close, but not far away. Nearsightedness, what you can see is the stuff that's near you. You can't see the stuff that's far away. For my friends who are nearsighted though, I like to joke that I am so myopic, my eyeballs are so myopic that I'm just like not sighted. Like without my glasses, I can't see anything. I've got really bad myopia. So nearsightedness, theoretically, You can see things that are close to you. You cannot see things that are far away from you. So when we talk about hyperopia, this is going to be the opposite of that, hyperopia. Hyperopia is going to be farsightedness. Farsightedness. If someone is farsighted, they can see close, but not far away. Or excuse me, they're farsighted. They can see far away. I'm saying it wrong. Sorry. They can see far away, but not close up. Now, I want to mention something because in a moment, we are going to talk about another condition that's similar to hyperopia called presbyopia. Presbyopia is what we would call age-related farsightedness. It's the fact that as people get older, they're more likely to not be able to see things up close and just see things far away. Hyperopia is when you are farsighted starting in your childhood. This is something that's kind of always around. It's not because you hit age 35 or older. It's because you just generally have hyperopia. When we talk about myopia and hyperopia, these are conditions that, like I said, can start in childhood and will continue throughout a person's life. The reason that these two conditions start in childhood and continue throughout a person's life is because it has to do with the shape of the eyeball. So notice in myopia, in nearsightedness, a person who has myopia, their eyeball is too long. So when we have someone with myopia, like Dr. Ollis, here come the light waves coming into the eyeball. The cornea bends them to get them to the lens. The lens bends them to send them to the place where normally, if the eyeball was the right shape, this is where the retina would be. Except the eyeball is really long. The eyeball is too long. My retina is all the way back here. My light waves get focused in front of it. And remember what we said, if I focus my light waves not on the retina, can I see them clearly? If I'm focusing them not on the retina? Yeah, remember our underline highlight star statement about vision, we've got to focus things on the retina. In someone with myopia, we send our light waves in front of the retina. We're not going to perceive them. They're not going to be clear. The problem with myopia is the retina is back farther than the lens thinks it is. So the lens doesn't send the light waves all the way back to the retina. It focuses them in front of the retina, which means that the retina can't see them clearly. So someone with myopia, we're going to use what's called a concave lens. Now, I promise I'm not going to ask you about that on the assignment or on the exam. I just want to point out for you that the shape of the lens matters. Notice how we kind of cave in that lens. The goal of a concave lens is to make those light waves travel farther in the eyeball to get them back to the very back where the retina actually is. So when I was a kid growing up, my optometrist told me that my eyeball was shaped more like a football than like a soccer ball because my eyeball was too long. So in myopia, in nearsightedness, where you can see things up close, but you can't see them far away, this is an issue with sending those light waves in front of the retina. Or in other words, the retina is too far back. I can't focus my light waves on it without using some kind of lens to make those light waves travel farther in the eyeball. Let's compare that with hyperopia. Again, hyperopia is a condition that develops in childhood. So it's something that's happening all of a person's life. With hyperopia, the eyeball is too short. So light waves come into the eyeball, the lens focuses them where the retina normally would be, except the retina is closer to the front of the eye. So those light waves are not being focused on the retina, they're being focused behind it. Since I am sending those light waves behind the retina, can I see clearly if I send those light waves behind the retina? Yeah, again, this is another situation where they are not being perceived clearly because we're not focusing them on the retina. In hyperopia, the issue is that my eyeball is too short, or in other words, my retina is closer to the front of the eye than my lens expected it to be. My lens sends it back to the normal position. The retina is not there. Because the retina is not there, I can't perceive those light waves. So check this out. Someone who has hyperopia is going to use what's called a convex lens. A convex lens is a different shape. Notice how it bulges. The job of a convex lens is to make the light waves focus closer in the eyeball, or to pull them more toward the front. So we'd use a convex lens to help those light waves not travel quite as far in the eyeball to get them to where the retina is, which is closer to the cornea. In someone with myopia, we're going to use a concave lens. That concave lens helps those light waves to travel farther to get back to where the retina is. These kinds of comparisons that I've been mentioning to you. So things like I can see close, but not far away. Or I can see far away, but not close. Or my eyeball's too long, it's too short. Those are the kinds of comparisons you're going to see that show up on the homework assignment. So make sure as you're studying our vision disorders, make sure you can describe these disorders. When we wrapped up the unit number 3 exam, I asked you all what were things that helped you prepare for it. And a lot of you said that talking out how things worked was really helpful for you. This would be an important thing for us to talk out, how these things work. Okay, so Jacqueline has a question. Okay, so Jacqueline's question is asking about why you can see things up close versus far away when it's based on the size of the eyeball. It kind of comes back to what we talked about here with the way that we have to modify light waves. When we're looking at something that's up close to us, the angle that the light waves come in at, we're going to bend them a little bit differently than when we have to bend things when they're coming in from a distance, at a farther angle. So it has to do with the mechanics or the physics of how the light waves get into the eyeball. I'm not going to ask you specifically about that. I know there are a couple of videos in the guided lesson that might more specifically address your question, Jacqueline, but the short answer to it is all of my light waves have to get bent into the eyeball. When I start changing the distance that they come in, not everybody's coming in straight. Some are coming in this direction, some are coming in straight. The angle that they come from changes what makes it harder or easier to focus stuff. Yes, yeah, so Christina mentioned if your eyeball is too long, we just can't travel light back to the right place. Yeah, we have a really hard time getting light back to the right place if it came from a distance. That's exceptionally hard for us. Whereas over here, It's harder for us to grab those light waves that are coming in from up close. Their angle is just too hard for us to bend them to here. Yeah, absolutely. Okay, so let's talk about a related vision disorder. This related vision disorder is called presbyopia. Presbyopia. My easy name for presbyopia is age-related farsightedness. Age-related farsightedness. Or what I like to joke about when we're talking about presbyopia is this is the reason everyone needs reading glasses. Reading glasses. Do I have any friends in class today who have reading glasses? I'm afraid my time is about to come. It's real sad to me. Age-related farsightedness. Yep, yeah, your mom, my grandma. Pretty much, honestly, pretty much everyone is going to develop presbyopia over the course of your lifetime. Let's remind ourselves about this. Presbyopia is age-related farsightedness. If I say that you're farsighted, what does that mean? If someone is farsighted, what can they or can't they see if they're farsighted? Exactly. So we can see things that are far away. Far away equals clear. Things that are up close, so things like books, newspapers, cell phones, all of that is going to be blurry. Everything that is close to you in presbyopia, you can't see it clearly. Let's talk about presbyopia. Well, actually, before we do, let me ask you a question. When we talk about hyperopia, which was also farsightedness, when we talked about hyperopia, what was wrong in hyperopia? Why could we not see things up close? What was wrong with the anatomy of the eyeball in hyperopia? Yeah, so in hyperopia, the length of the eyeball was too short. So eyeball too short in hyperopia. Because the eyeball was too short, the lens was sending the light behind the retina. So lens sends light too far back. So now we're talking about another version of farsightedness. In presbyopia, in age-related farsightedness, it's not that the eyeball is too short. The eyeball shape is just fine. The problem is I can't get the eyeball to, or I can't get the light focused on the retina. I'm still sending that light too far back in the eye. It's not because the eyeball is the wrong shape. It's because I've got the issues listed right here. The first problem that I have in presbyopia has to do with my lens. Remember that we said the lens gets short and stout or long and skinny to help us focus light. So when we talk about the lens in presbyopia, in age-related farsightedness, the problem that we're having is it's calling it its decreased power of accommodation. Let's just put this in easy person words. In easy person words, the lens gets hard. In easy person words, the lens gets hard. It gets stiff. My power of accommodation is the fancy way of saying changing size. Unfortunately, the older we get, the harder our lens becomes. The less accommodating to changing sizes it becomes. So problem number one in presbyopia is my lens is getting hard. I'm having a hard time doing anything to change its shape. Problem number 2 is the ciliary body or the ciliary muscles. As we get older, the ciliary muscles get weaker. So the ciliary muscles get weaker. Remember that I needed the ciliary muscles to change the shape of the lens. When I flexed or when I contracted these ciliary muscles, that made my lens go short and stout. So let's make a note for ourselves here. Contract muscles, short and stout lens. A short and stout lens helps me to see things up close. When I squeeze down my lens and I make it smaller, that helps me to focus the waves of light that are coming from close to me. That's the way that up close vision works. Squeeze the muscles, squeeze the lens, I can see things up close. As we get older, the lens gets harder and the muscles that are supposed to squeeze it into a new shape get weaker. When those muscles get weaker, they have a harder time pressing on that lens, getting it to squeeze down. And I need it to squeeze down for me to be able to focus my light on the retina. So as a person ages, their ability to see things up close goes down because we can't take those light waves that come in at such a sharp angle. I can't squeeze them into the right shape because my lens is too hard and my muscles are too weak. I can't get that light focused on the retina. Presbyopia, an issue with the lens and the ciliary body. Remind me again, what was my issue with hyperopia? What happened in hyperopia? What was my problem? Yeah, we've got that short eye, right? I still can't focus my light on the retina. I can't, my eyeball's too short. In myopia, what was my problem? What happened in myopia? The other one we didn't list on here. Yeah, in myopia, the eyeball's too long. Eyeball too long. We still have the problem, though, that in myopia, light waves still aren't getting focused on the retina. Just like they're not getting focused on the retina in hyperopia, just like they're not getting focused on the retina in presbyopia. So again, that big picture idea that I told you all to keep in mind, for us to be able to see things, we have to focus the light on the retina. If it doesn't hit the retina because the retina is in front of where we expected it to be, or if it doesn't hit the retina because the retina is farther back than we thought it would be, or if it can't get there because we can't bend the light waves to where it is, it doesn't matter. It always ends up in the wrong place because we're not getting it to the retina. Let's talk about one more vision disorder here. This one more vision disorder to mention is what we call astigmatism. Astigmatism. Does anybody know that they have astigmatism? Okay, one person. I have a mild astigmatism. For those of us with eye disorders, it's like you just get them all, right? I don't have presbyopia yet, but my time is coming. Okay, I want to stop talking for a moment because I want you all to help me in the chat. Can we figure out what's going wrong with astigmatism? What's the problem if someone has astigmatism? Yeah, so Nicole is voting for the cornea, and that's correct. We've got an issue with the shape of the cornea. If someone has astigmatism, instead of the cornea being a sphere, like we see here on the front part of the normal eyeball, that cornea is a little bit more oval shaped, meaning it kind of sticks out a little bit more. It's a little more oval. If you remember when we started talking about the process of vision, I told you that the cornea was the first place that we changed the direction of light waves. So the cornea is really important to help us with focusing light. If the cornea messes up the way that light is focused, the lens is not going to be able to fix the problems that the cornea did. So if someone has astigmatism, when light first comes into the eyeball, the cornea doesn't bend it correctly. The lens tries to focus it, but because it's coming in at such weird angles, we end up with, like a couple of us said in the chat, multiple focal points. Meaning I send that light, or I try to focus that light in two different places. Notice on the example in our picture here, one of those places is in the vitreous humor. One of those places is behind the eyeball. So neither of these focal points is actually on the retina, which again, true or false? Light needs to be focused on the retina for me to be able to see it. True or false? Light needs to be focused on the retina to see it. Yeah, several of us are chiming in. I must get it focused on the retina for me to be able to see. When we have astigmatism, we're going to focus that light in more than one place. Probably none of those places we're focusing it is going to be the retina. So again, we cannot see clearly. So I'm going to steal a little bit of my thunder for Monday's group work, but here's what I think that you should do if you haven't already done this in your notes. I think that you should make a table that compares each of the vision disorders. So what we need to know for each of these vision disorders is the anatomy. Essentially, which part of the eyeball is the wrong shape. We need to know when the disorder develops. Is this something that a person is born with? Is this something that they develop when they're older? We should also be able to describe what happens to the light. So we should know where that light's going. Is it in front of the retina? Is it behind the retina? Is it in more than one place in the eyeball? We will take some group work time on Monday to review some of the things like this. You know how Dr. Allis is though, right? I do love me some compare and contrast questions. We have 4 vision disorders that we're learning about. And you better believe I'm going to make you compare and contrast them. And the homework assignment makes you do that too. So work on figuring out how these disorders are the same and how these disorders are different. That's a really important comparison to make. Before I move into more of the micro anatomy or the up close anatomy of the eye, do we have any questions at the moment? I'll send you all an eyeball. How do we feel about the disorders? I'm sure we're still processing. It's totally fine. Okay. Well, let's move into the process of vision a little bit more in depth. We're going to talk about the way that the cells of the eyeball work. So in the eyeball, we have or in the retina, more specifically, maybe more specific. In the retina, we have cells called rods, and we have cells called cones. Rods are my type of cells. They're just kind of like your everyday vision cells. These ones are really good at seeing the difference between light and dark. You might call them black and white vision cells. When we talk about seeing in color, the cells that help us to do that are called cones. And we have different kinds of cones in the eyeball. The different kinds of cones in the eyeball each detect different types of wavelengths of light. Now, again, let me mention that I am not going to ever ask you these numbers that are on our graph here. So don't worry too much about these numbers. I am including this to show you how the process of color vision works. In your eyeball, we have what are called blue cones, green cones, and red cones. And that kind of gives you an idea of the general colors that these types of cells detect. If I am a red cone, the type of light wave that's most exciting to me is something that travels at a speed of 560 nanometers. That's the top speed, or the speed that I really detect super well. But I can also detect light waves that are traveling at a wavelength of 700 nanometers. And I can detect wavelengths that are traveling that are as long or as short as 475 nanometers. The types of wavelengths that activate me, that help me detect something happening, here's my range if I'm a red cone. If I'm a green cone, the types of wavelengths that I detect are a little bit shorter, so over here maybe in more of the purple range, going up through about orange. I can't really detect the red waves, but up through about orange I can detect those. If I'm a blue cone, I can definitely detect those shorter wavelengths, so the purple, blue, but as we get closer to kind of yellow and red, I can't detect those colors anymore. For you to perceive a color in the world around you, let's say that wavelengths are coming in, we'll do right here at our mixing point here. Say I've got some wavelengths of light that are coming in at about 550 nanometers. So we're going up and down at 550. That comes in and it activates a green cone to some extent, and it also activates a red cone. The way that you figure out what color you're seeing is by adding together how activated each of these different kinds of nerve cells are. What was the name of the process that we use when we are adding together multiple messages to figure out what's happening or to interpret what's happening? Yeah, so individual neurons do the process of summation. They add messages. And if I take these sensory neurons and I send them to the brain, what was the word for what we said the interneurons in the brain do? What's that called when they do processing? Yeah, integration. So integration and summation, both of those things. So integration or summation, the way that you figure out what color you're seeing is by listening to what the green cones are saying. So they're saying, I've absorbed about 90% of this light. And then we listen to what the red cones are saying. They're saying, I absorbed about 90% of this light. And then we listen for what the blue cones are saying. And the blue cones are saying, I absorbed 2% of this light, for example. We add together those messages from the three cones, and that helps your brain to know what color you're seeing. So say we've got some more light waves that come in over here, and my red cones are saying, I absorbed 25% of this light. And the green cones are saying, I absorbed 10% of this light. And the blue cones are like, what light? I don't know what you're talking about. Your brain adds those messages together to figure out, oh, we're seeing something that's orange, for example. So the way that color vision works is we listen to all three types of cones and we add together, or we integrate their messages together to figure out which color on the spectrum we're seeing. So an important idea about cones is that we have three different kinds of cones. The types of proteins that they use to detect color are different in each of them. That's why they each detect different colors. When we talk about rods in your eyes, remember we said rods are kind of black and white vision. When we talk about rods, there's only ever one kind of protein that they use to detect things. That one kind of protein, it detects light best at about 500 nanometers, but it'll detect light of all of these different lengths as well. But the thing with rods is they don't listen to more than one type of cell. With rods, it's kind of like all or nothing. Either you're being activated or you're not being activated. So they do pretty well with most of the colors on the spectrum, but they're not going to have the specificity to tell you, oh, we're seeing something that's orange, as opposed to we're seeing something that's green. Here's a good table, an underline, highlight, star, must know table when we're talking about the process of vision. It's a comparison for us of those rods versus those cones. So let's go through and talk about the differences between them. When we talk about rods, rods are the ones that I told you they detect light at one frequency. There's only one version of a rod. When we talk about cones, we had those three versions. We had red cones, blue cones, green cones. So that's going to lead them to see either in color, if you're a cone, or non-color. It's just black or white with a rod. They see black and white. Even though rods can't tell you what color you're looking at, the benefit of a rod is that they're highly sensitive. Highly sensitive means it doesn't take much light for this type of cell to be activated. So when it's really dim, when it's the middle of the night and you're getting up to feed a baby, or maybe that's just me, right? Well, when you get up in the middle of the night to get a drink or go to the bathroom, the kind of cells that can see a little bit are these rods. Those cells are highly sensitive. Any light in your environment, they will detect it. Compare that to cones. Cones are low sensitivity. Low sensitivity meaning that you're going to need to have a lot of light for these cells to be able to detect it. So if a cell is low sensitivity, they're not very sensitive. They're not going to detect things very easily. You need a lot of it. If we're a highly sensitive cell, it doesn't take much. We can be activated. When we talk about rods, we have a lot of them. They're more numerous than cones. So there's a ton of this type of cell in your retina. But the big idea with rods is that they're found in the peripheral retina, meaning they're not found in the macula ludia, the place where we focus light on the retina. We don't have any rods there. We just have cones there. But tons and tons of rods all over the retina, functioning at high sensitivity, meaning they can detect stuff all over the place, but they're just going to tell you black and white. They're not going to tell you if it's colored. Cones are found throughout the retina, but they're most concentrated in the very middle. They're most concentrated in the place where your eyes normally focus light. And think about it, that kind of makes sense. I need a lot of light to turn on a cone, to activate a cone. So I'm going to put them in the place where my light usually goes. Even though I have three types of cones, I don't have as many of them in the eyeball. So I really focus them in the center of the retina, and that's where my light, my bright light, is going to go to activate them. That's how I see in color. A fun experiment that you can do with rods versus cones. When you're outside this evening at like, what is it these days, like 5, right? When it starts to get dark, take a look around you Notice that when it is legitimately getting dark, when we're at twilight and when it's getting darker, notice that you can actually see things more clearly in your peripheral vision, so the stuff that's beside you, as opposed to what's straight in front of you. Because what's straight in front of you, you're trying to detect with cones. If there's not a lot of light, those cones aren't being activated. What's straight in front of you, you can't really see anything. But the stuff that's in your peripheral vision, the stuff that's kind of in the outside vision, that's where the rods live. And when we have rods, they are highly sensitive. They can pick stuff up when there's not a lot of light out there. So what you'll see in your peripheral vision is going to be more clear than what you see straight in front of you. And the other thing, though, you can remember back to a time you were out when it was starting to get a little bit dark. Take a moment to recognize that what you see when it's starting to get dark is just black and white, because the cells that would help you see in color aren't picking up enough light to tell you what color things are. So it's not like the color of your car changes at nighttime, but when it starts to get dark, it's harder for you to see that color, because the only cells that are working, or that are working really well, are the rods, and they just see in black and white. So rods and cones, must be able to compare those to each other. Hyperopia, presbyopia, myopia, astigmatism, need to be able to compare those things to each other as well. I think I want to pause there for today, because I would hate to start the process of hearing and not be able to wrap it up. I don't want to leave us hanging on it. So I'm done with everything I want to cover today. What questions do we have about vision or senses or unit number 4? I'll give us our little cricket here. Yes. So I will work on making, Jacqueline asked about a Unit 4 review assignment. Yes, let me put that on my list as well. I will work on making one of those too. So we'll get a Unit number 4 review assignment up, and then I will also work on getting a final exam assignment up for you too, to help you practice. Yeah, Christina asked if we needed to know about how to treat the disorders. I'm not going to ask you specifically, is it a concave lens or a convex lens? That's for your physics class. That's not for me. I mainly point that out to say that when we're looking at these types of vision disorders, I have to address them differently, because I have to move my light in different ways. So if I need my light to travel farther, I'd have to use a concave lens. If I need it to travel less far, I'm going to use a convex lens. Hey, let's do a tie-in. Our picture doesn't specify for sure. When someone has presbyopia, they have to use reading glasses. Would reading glasses be convex or concave? Can we make a prediction? If someone has to have reading glasses because they have age-related farsightedness, yeah, they're going to have those convex lenses, just like a person with hyperopia. Although actually, more accurately, probably someone with reading glasses is going to have bifocals, possibly, where the top half could either be concave or it just might not be glasses at all. And the bottom half would be convex, where we're bending those light waves, getting them closer to the front of the eyeball. So since the vision problem is the same, since we're sending those light waves, let me go back here. Since we're sending those light waves too far, I would use the same kind of intervention on them. So I would treat it the same way that I treat hyperopia. But again, I'm not going to get that specific in the weeds for you. That's just a fun fact. Yeah, Nicole asked about treating astigmatism. Yes, astigmatism would be another thing that we would need to do lenses for. The thing with astigmatism as well is the shape of the cornea can be very different from person to person. So what we kind of do, at least when you think about something like, for all my friends who wear contacts, there's a special kind of contacts called toric lenses. Toric lenses, if you have a very severe astigmatism, you have to use this special kind of contacts. Basically, the goal of a toric lens, or if you have glasses to treat it, is we kind of have to almost redo what's going on with the cornea. So redo the shape of the cornea, put something that works more powerfully than the cornea does to bend light the correct way. So short answer to your question, Nicole, yes, we would use something like that. Hey, we didn't talk about this. There is a disorder where that would be similar to presbyopia, and that is an issue with the lens. Your lens can become cloudy and make it hard for you to see through. Does anyone happen to know the name of the vision disorder when your lens gets cloudy? Yeah, so when the lens gets cloudy, that's called glaucoma. Or not glaucoma, sorry. Glaucoma is, I'll talk about glaucoma. It's called cataracts. I read your answer too. When the lens gets cloudy, that's cataracts. Lens gets cloudy. With glaucoma, that is an issue with the blood vessels in the eye. When the blood vessels and the blood pressure in the eye get too high, that can lead to what you call glaucoma. So glaucoma has to do with the pressure. When your lenses themselves get cloudy and it's hard to see through them, that's called cataracts. There are actually a couple of articles in the current events in anatomy options for you with Unit 4. There's so many articles for Unit 4 because special senses is just like amazing. But one of the articles in the Unit number 4 current events talks about eye drops to help make your lens less cloudy. So if you're interested in reading about that, there is a treatment. They're mainly using it, I think, right now experimentally, but a treatment to help make your lens get less cloudy by putting eye drops on the outside of the eyeball. So very fascinating there with the cataracts. Yeah, so Christina asked a question in the chat comparing presbyopia to hyperopia. Her question is that the biggest difference between them has to do with age, and that is correct. So it has to do with age when they're developed. We still have the same problem with the focusing of the light on the retina, but the other difference that I'll mention is what causes these disorders. What was it with presbyopia? What happens as a person ages that makes us have issues focusing the light? Can we summarize why we focus light in the wrong place with presbyopia? Yeah, so with presbyopia, the reason I focus light in the wrong place is because my lens is hard, because I'm getting older, and my ciliary body is weak. So my light ends up getting focused behind the retina. It's going too far. In hyperopia, I don't have issues with my ciliary muscles or my lens. What's the issue in hyperopia? What's giving me trouble in hyperopia? Exactly. It's all about the eyeball. So the eyeball shape is wrong. That means that light goes behind the retina. That's hyperopia. In presbyopia, The eyeball shape is fine, but the things that are supposed to cause me to focus my light on the right place in the eyeball, I can't use those. So in both of them, we send light behind the retina. One, because the eyeball is too short. One, because the lens is too hard and the muscles are too weak. So age is a difference, and the actual anatomical cause of the disorder is different as well. Gloria asked about when hyperopia develops. Yeah, that can definitely be something that's present at birth. I was chatting with one of my many optometrists over the years, and he was talking about how his daughter had severe hyperopia back when she was a baby, and she had the hardest time kind of picking up toys and figuring out how to get them like to her mouth or to her face because she was so severely farsighted. So yeah, that can be something that starts really young. Typically, if you see young kids in glasses, it's probably hyperopia that they're treating, that the eyeballs are still a little bit short, that they need to continue to grow. Yeah, hyperopia and myopia, I believe probably we would consider those both genetics. Yeah. And there's actually a very interesting, again, current events in anatomy, keep referencing back to it, but there's some articles, they've done a lot of studies in China, for example, where firstborn children, granted a lot of families in China just have one child, but they were trying to speculate, is it because, what are some of the reasons that would contribute to this high rate of myopia in our children? So there's another article that you could read about that. So yeah, so myopia can happen at birth, definitely, Gloria. I think the thing with myopia is it's harder to catch younger, just because like you don't, it's not going to completely impair your ability to function if you can't see far away as dramatically as not being able to see up close would. So myopia, you have to have the wherewithal and the understanding to realize that you can't see things far away. For me, it was when I started school. First grade is when I got my first set of glasses. Gotta love that. But chances were my eyeballs were already too long, so I just didn't know it. But yeah, myopia and hyperopia are the two that you're kind of born with, and over time they can get worse as the size of your eyeball changes when your head grows or when your body grows. Yeah, Gloria feels me with the young glasses. Yeah, Christina mentions doing some extra credit. Definitely do some extra credit. There's a ton of really cool articles about special senses. So take advantage of it this unit for sure. All right, well, I'm going to call it a day for today. Plan Monday morning. We're going to pick up and keep going. We'll do the senses of smell, or excuse me, the senses of hearing and balance for sure Monday morning. And then probably try to knock out Monday night the senses of taste and smell. Those two are super simple compared to what we're about to hit with hearing and vision. So we can look forward to that ending on an easier note. But don't forget about lesson number 12 assignment due by tomorrow night, Friday night, and start working on lesson number 13 as well. So have a great afternoon. I hope to see a bunch of you on Monday.